Hello, my name is YeonwooPark, and I'm a graduate student at the University of Chicago. I am grateful for this opportunity to present my work on historical memory, epistasis, and contingency in long-term protein evolution. A fundamental question in evolutionary biology is the extent to which evolution is contingent on historical path taken in the past. There are many possible causes of historical contingency, one of them being epistatic interactions among mutations within a protein. When mutations within a protein interact with each other, the phenotypic effect of a mutation can change during evolution depending on what other mutations fix. For example, consider two mutations and their phenotypic effects. Here, mutation 1 decreases the phenotype, while mutation 2 has no effect. When there's an epistatic interaction, mutation 1 may increase the phenotype when preceded by mutation 2. The converse is also possible where mutation 1, which increases the phenotype on its own, has a negative effect when preceded by mutation 2. In this way, evolution can be contingent on what epistatic modifiers were present in the mutational path taken in the past. A central task in understanding the extent to which evolution is historically contingent is therefore to reveal the extent and impact of epistasis during evolution. The frequency and magnitude of epistatic interactions during evolution are unknown. This is true despite the recent surge of deep mutational scan studies that measure the interactions among thousands of mutations. These studies report epistasis among the many possible mutations, but do not directly show how mutations that fix during evolution change the effects of other mutations. Ancestral reconstruction studies can directly reveal the historical changes in the effects of mutations, but have been limited to only a small fraction of possible mutations. What we need to know is how substitutions sampled during history change the effects of other mutations and modulate their evolutionary fate. To answer this question, we must trace the change in each mutation's effect along reconstructed evolutionary trajectories. Before describing the experiments, I will introduce a conceptual framework that we developed for characterizing the extent of epistasis. First, consider a scenario when there is no epistasis. This plot shows the effects of two mutations during evolution. Under no epistasis, a mutation's effect will remain constant throughout evolution. An extreme opposite scenario is when epistasis is pervasive. In this case, a mutation's effect will change dramatically by each substitution and be made completely unpredictable. An intermediate scenario is also possible, where a mutation's effect changes, but not as dramatically. After some amount of time, the mutation's effect will change by a sufficient magnitude to be uncorrelated from the initial effect. We call that amount of time the memory length of the mutation. The three scenarios imply very different evolutionary dynamics. In the first, the evolutionary fate of a mutation is permanently dictated by the initial effect. In the third, it is completely contingent on the most recent substitution. In the middle, it is shaped by initial effect for some duration, then becomes contingent on the historical path taken. The memory length of a mutation is infinite, finite, and zero in the three scenarios. Our task is then to measure the memory length of every mutation in a protein. To do that, we traced how the effect of every point mutation changed during the evolutionary history of steroid receptor DNA binding domain. Steroid receptors are transcription factors distributed across animals. They mediate the actions of steroid hormones by binding to specific DNA response elements and regulating the expression of target genes. We inferred a phylogeny of steroid receptors and reconstructed the ancestor of all steroid receptors, the deepest blue node shown here, which existed in the last common ancestor of bilaterians. We also reconstructed 6 other ancestral proteins, leading to modern-day steroid receptors. This schematic phylogeny shows the reconstructed trajectory with the percentages showing the amount of sequence divergence across each interval. In total, we applied deep mutational scanning on 7 ancestral and 2 modern-day proteins spanning 42% divergence. We used a novel microarray-based method to construct all point mutants for the 9 DVDs. We then used fluorescence reporter assay and source-seq to characterize the phenotype of each mutant. In this assay, we transform a steroid receptor DVD into an E strain where DNA binding leads to GFP transcription. We quantify the GFP fluorescence as a measure of a mutant's DNA affinity. To do this for all mutants, we introduce the mutant library into a population of yeast cells such that each cell contains a unique DVD. By sorting the population into bins and sequencing the DVDs, we can infer the mean fluorescence of each DVD mutant. We performed 3 replicate sourcing experiments and obtained highly precise measurement for 99% of mutants. We defined the effect of a mutation, delta F, as the difference between the fluorescence of the mutant and wild-type DVD. The effect of each mutation is measured in 9 historical backgrounds. This plot shows an example effect trajectory for mutations serine to proline at site 9. The measurements are organized in relation to the phylogeny shown along the x-axis. We calculate the amount of change in a mutation's effect across each of the 8 phylogenetic intervals, denoted delta delta F. We first determined how common epistatic interactions were during evolution. To do so, we determined whether there are significant differences among the 9 historical effects of each mutation. For 40% of mutations, there were no statistically significant differences. For the remaining 60% of mutations, there are significant historical changes in effect. This shows that the effects of most point mutations change during steroid receptor evolution. We then measure the memory length of each mutation. To do so, we model the effect of a mutation as a random variable undergoing Brownian motion with sequence divergence. In this model, each mutation's effect changes at a characteristic rate denoted sigma. This plot shows how the effects of 2 mutations with small rate change during evolution. After a sequence divergence of D, the amount of change in effect will be distributed normally with variance equal to sigma squared times D. The bottom plot shows how the effects of 2 mutations with large rate change during evolution. We infer the rate of change in a mutation's effect from the 8 delta delta F values, each of which is an instantiation of Brownian motion. Once we infer the rate for each mutation, we group mutations with similar rate together. This plot shows how the effects of 5 mutations with similar rate can change during evolution. We compare the effects of these mutations at different time points. Here, each dot shows the effect of a mutation at time D0 versus D1. The effects at D0 and D1 are highly correlated. When we compare the effects at D0 and D2, we find that they are almost uncorrelated. We perform this comparison along our phylogeny to measure how quickly the temporal correlation in mutation effect decays. We model the correlation as an exponentially decaying function and measure the memory half-life, the amount of sequence divergence expected to halve the temporal correlation in mutation effect. This is our measure of memory length. We found that memory half-life varies dramatically among mutations. This is a histogram of memory half-life across all mutations. Memory half-life ranges from less than 25% sequence divergence to virtually infinite. We took mutations with short memory half-life and compared their effects between 2 proteins separated by 42% divergence. As expected, their effects are almost completely uncorrelated between the 2 proteins. By contrast, mutations with long memory have essentially identical effects on the 2 proteins. This shows that the number or strength of epistatic modifiers vary dramatically among mutations within a protein. In particular, 1/3 of mutations' effects are almost completely randomized during evolution. Next, we ask whether memory length is an invariable property of a mutation or varies from path to path. We have so far assumed that the effect of a mutation changes at a constant rate throughout evolution. This will happen if many historical substitutions interact with a mutation at similar strength. Alternatively, a mutation's effect may change at a variable rate or episodically in only some intervals. This will happen if the strength of epistatic interaction varies widely among historical substitutions. We can answer this question by testing how accurately the Brownian motion model, which assumes constant rate, describes the course of change in a mutation's effect. To do that, we re-infer the rate of change in a mutation's effect after excluding one phylogenetic interval. We then use the re-inferred rate and the Brownian motion assumption to predict the amount of change in the mutation's effect across the excluded interval. This plot shows the result of prediction for one interval. The red dots show the predicted distribution, and the gray bars the observed distribution. There's a remarkable agreement between the two, indicating the overall accuracy of the Brownian motion assumption. Because this plot shows the prediction for all mutations, it may be masking some model inaccuracies for individual mutations. So we divided mutations into 10 groups based on the predicted amount of change in mutation effect. This plot shows the result for the 10% of mutations with the largest predicted change. And this plot shows the result for the 10% with the smallest predicted change. We compared the predicted and observed standard deviation of delta delta F for each decile of mutations in each interval and found a very good agreement between the two. These results show that the effects of most mutations changed at a nearly constant rate throughout evolution. They indicate that memory length is a largely invariable property of a mutation. They also indicate that historical contingency arises gradually from accumulation of many small effect epistatic modifiers. Next, we asked how the memory length of a mutation shaped its evolution. We analyzed the impact of initial phenotypic effect on the fate of long memory mutations and short memory mutations. First, we calculated the effect of every mutation on ANKNR3, the initial ancestor in our phylogeny. Next, we selected only those mutations that fixed along the phylogeny and plotted the distribution of their effects. Compared to all mutations, they are highly enriched to a weak or positive effect, indicating that mutations with strongly negative effects were deleterious. We then made this comparison only for long memory mutations. As expected, we found the initial effect had a strong impact on the mutation's fate. Mutations with initially weak or positive effects are 30-fold enriched among substitutions than mutations with initially strongly negative effects. For short memory mutations, initial effect had much weaker impact. Mutations with initially weak or positive effects are only 3-fold enriched than mutations with initially strongly negative effects. The fates of short memory mutations were instead contingent on epistatic interactions. This is the historical effect of a mutation that fixed at the point marked by the arrow. This mutation was initially inaccessible, but was permitted by epistatic interactions. This mutation, on the other hand, was initially accessible and was contingent rather on the lack of restrictive epistatic interaction that prevented its fixation in other parts of the phylogeny. Overall, these results show that the fate of a long memory mutation is dictated by the initial phenotypic effect. By contrast, the fate of a short memory mutation is highly contingent on how it interacts with the substitutions along the particular historical path taken. Finally, we asked how the initial phenotypic effect and memory length together shaped sequence evolution. We examined the frequency of 3 types of mutations: those with short memory, those with long memory and are initially accessible, and those with long memory and are initially inaccessible. These mutations are nearly equally abundant. Historical substitutions are, however, dominated by long memory, initially accessible mutations. Short memory mutations are depleted 7-fold compared to long memory, initially accessible mutations. This implies that transient incompatibility with the genetic background frequently restricts short memory mutations from fixing. This then implies that analyzing only the mutations that fix during evolution underestimates the contingent nature of sequence evolution because epistasis frequently leads to loss of mutational opportunities. In conclusion, we found widespread and strong historical contingency where 1/3 of mutations' effects are completely randomized during evolution. We found that contingency arises gradually from accumulation of many small effect epistatic modifiers, and that the degree of contingency varies dramatically among mutations within a protein. Predicting a mutation's evolution requires knowing its memory length, which can only be learned from comparison of mutation effects within a phylogenetic framework. With that, I would like to end by acknowledging the immense encouragement and stimulation from our lab members that made this project possible. Thank you very much.